Dear students, today we are going to discuss the topic range of activity and mechanism of action of antibiotics, which include sulfa drugs, penicillin, aminoglycosides, quinolones and cephalosporins. The main objectives of the topic are to understand what antibiotics are, to identify the major types of antibiotics and their proper usage, define therapeutic effects, side effects and administration routes of major antibiotics, the mode of action of antibiotics along with their range of activity. Now, coming to the history of antibiotics, antibiotics revolutionized medicine in the 20th century and have, together with vaccination, led to the near eradication of diseases such as tuberculosis in the developed world. This effectiveness and easy access led to overuse, especially in livestock raising, prompting bacteria to develop resistance. This has led to widespread problems with antimicrobial and antibiotic resistance, so much as to prompt the World Health Organization to classify antimicrobial resistance as a serious threat that's no longer a prediction for the future. It is happening right now in every region of the world and has the potential to affect anyone of any age in any country. The era of antibacterial treatment began with the discovery of irreversible, first synthesized by Alfred Berthem and Paul Ehrlich in 1907 and used to treat syphilis. The first systematically active antibacterial drug Protosil was discovered in 1933 by Gerhard Domag, for which he was awarded the 1939 Nobel Prize. Coming to the introduction about antibiotics. An antibiotic is a drug that kills or slows the growth of bacteria. Antibiotics are one class of antimicrobials, a larger group which also includes antiviral, antifungal and antiparasitic drugs. Antibiotics are chemicals produced by or derived from microorganisms such as bacteria and fungi. The first antibiotic was discovered by Alexander Fleming in 1928 in a significant breakthrough for medical science. Antibiotics are among the most frequently prescribed medications in modern medicine. Some antibiotics are bactericidal, meaning that they work by killing bacteria. Other antibiotics are bacteriostatic, meaning that they work by stopping bacteria from multiplying. Each different type of antibiotic affects different bacteria in different way. For example, an antibiotic might inhibit a bacterium's ability to turn glucose into energy or its ability to construct its cell wall. When this happens, the bacterium dies instead of reproducing. Some antibiotics can be used to treat a wide range of infections and are known as broad-spectrum antibiotics. Others are only effective against a few types of bacteria and are called as narrow-spectrum antibiotics. This was a brief introduction about antibiotics. Now, let's discuss their range of activity. Depending on the range of bacterial species susceptible to these agents, antibacterials are classified as broad-spectrum, intermediate spectrum or narrow spectrum. The spectra of activity may change with acquisition of resistance genes. Now, let's discuss broad-spectrum antibacterials. Broad-spectrum antibacterials, they are active against both gram-positive and gram-negative organisms. Examples include tetracyclines, phenols, fluoroquinolones, third generation and fourth generation cephalosporins. Now, let us discuss the difference between gram-positive and gram-negative bacteria. Gram staining is a special technique which is used to stain bacteria. This technique was developed by Christian Gram in 1884. The stain used in gram staining is called gram stain. Chemically, gram stain is a weakly alkaline solution of crystal violet or gentian violet. On the basis of cell wall structure and its stain ability with gram stain, bacteria are grouped into two categories, gram positive and gram-negative. Gram-positive bacteria are the bacteria which remain colored with gram stain even after washing with alcohol or acetone, whereas gram-negative bacteria do not retain the stain while washed with alcohol or acetone. Outer membrane in case of gram-positive bacteria is absent, whereas the same is present in case of gram-negative bacteria. Cell wall is 20 to 30 nanometer thick in case of gram-positive bacteria, whereas it is only 8 to 12 nanometer thick in case of gram-negative bacteria. Lipid content in cell wall is very low in case of gram-positive bacteria, whereas in case of gram-negative bacteria, the lipid content in the cell wall is 20 to 30%. Cell wall contains teichoic acids in case of gram-positive bacteria, whereas teichoic acids are absent in case of gram-negative bacteria. A few pathogenic bacteria belonging to gram-positive group, for example, Corynebacteria and Staphylococcus. Most of the pathogenic bacteria belonging to gram-negative group, for example, E. coli, Campylobacteria, Treponema palladium. Now, let us discuss narrow spectrum antibacterials. These narrow spectrum antibacterials have limited activity and are primarily only used against particular species of microorganisms. For example, glycopeptides and bacitracin are only effective against gram-positive bacteria, whereas polymyxins are usually only effective against gram-negative bacteria. Aminoglycosides and sulfonamides are only effective against aerobic organisms, while nitroimidazoles are generally only effective for anaerobes. Now, let us discuss the mechanism of action of these antibiotics. These antibiotics work by inhibition of the cell wall synthesis, inhibition of protein synthesis, alteration of cell membranes, inhibition of nucleic acid synthesis, and antimetabolite activity. Different antibiotics have different modes of action owing to the nature of their structure and degree of affinity to certain target sites within bacterial cells. First, inhibitors of cell wall synthesis. While the cells of humans and animals do not have cell walls, the structure is critical for the life and survival of bacterial species. A drug that targets cell walls can therefore selectively kill or inhibit bacterial organisms. Examples are penicillins, cephalosporins, bacitracin, and vancomycin. Second is the inhibitors of cell membrane function. Cell membranes are important barriers that segregate and regulate the intra and extracellular flow of substances. A disruption or damage to the structure could result in leakage of important solutes essential for the cell survival. Because this structure is found in both eukaryotic and prokaryotic cells, the action of this class of antibiotics are often poorly selective and can often be toxic for systemic use in the mammalian host. Most clinical usage is therefore limited to topical applications. Examples are polymyxin B and colistin. Third, inhibitors of protein synthesis. Enzymes and cellular structures are primarily made of proteins. Protein synthesis is an essential process necessary for the multiplication and survival of all bacterial cells. Several types of antibacterial agents target bacterial protein synthesis by binding to either the 30S or 50S subunits of the intracellular ribosomes. The activity then results in the disruption of the normal cellular metabolism of the bacteria and consequently leads to the death of the organism or the inhibition of its growth and multiplication. Examples are aminoglycosides, macrolides, lincosamides, streptogramins, chloroamphenicol, and tetracyclines. Fourth is the inhibitors of nucleic acid synthesis. DNA and RNA are keys to the replication of all living forms, including bacteria. Some antibiotics work by binding to components involved in the process of DNA or RNA synthesis. which causes interference of the normal cellular processes and which will ultimately compromise bacterial multiplication and survival. Examples are quinolones, metronidazole, and rifampin. Fifth is the inhibitors of other metabolic processes. Other antibiotics act on selected cellular processes essential for the survival of the bacterial pathogens. For example, both sulfonamides and trimethoprim disrupt the folic acid pathway, which is a necessary step for bacteria to produce precursors important for DNA synthesis. Sulfonamides target and bind to dihydropteroate synthase trimethoprim, inhibit dihydrofoliate reductase. Both of these enzymes are essential for the production of folic acid, a vitamin synthesized by bacteria but not humans. Now, let's discuss the types of antibiotics. Although there are several classification schemes for antibiotics based on bacterial spectrum, that's broad versus narrow, or type of activity, that's bactericidal versus bacteriostatic. The most useful is based on chemical structure. Antibiotics within a structural class will generally have similar patterns of effectiveness, toxicity, and allergic potential. The main classes of antibiotics are penicillins, aminoglycosides, quinolones, sulfonamide, and cephalosporins. While each class is composed of multiple drugs, each drug is unique in some way. First, let us discuss penicillins. The penicillins are the oldest class of antibiotics. Penicillins have a common chemical structure which they share with the cephalosporins. Penicillins are generally bactericidal, inhibiting formation of the bacterial cell wall. Penicillins are used to treat skin infections, dental infections, ear infections, respiratory tract infections, urinary tract infections, and gonorrhea. The natural penicillins are based on the original penicillin structure. Penicillin G types are effective against gram-positive strains of streptococci, staphylococci, and some gram-negative bacteria such as meningococcus. Penicillin are resistant penicillins, notably methicillin and oxacillin, are active even in the presence of the bacterial enzyme that inactivates most natural penicillins. Aminopenicillins such as ampicillin and amoxicillin have an extended spectrum of action compared with the natural penicillins. Extended spectrum penicillins are effective against a wider range of bacteria. Extended spectrum penicillins are piperacillin and ticarcillin. Penicillins and their side effects. Penicillins are among the safest and least toxic drugs. The most common side effect of penicillin is diarrhea. Nausea, vomiting, and upset stomach are also common. In rare cases, penicillins can cause immediate or delayed allergic reactions which manifest as skin rashes, fever, angioedema, and anaphylactic shock. Severe hypersensitivity reactions are more common after injections than after oral formulations. Neurotoxicity, that's very high dose of penicillins, especially in patients with renal impairment, may cause convulsions. Penicillins are classified as pregnancy category B. Now, cephalosporins. Cephalosporins and penicillins are closely related, but cephalosporins are isolated from another group of microorganisms, Cephalosporium. Cephalosporins differ from penicillin by having a 6-membered heterocyclic sulfur-containing ring attached to the 4-membered beta-lactam ring. The first cephalosporin isolated from Cephalosporium was cephalosporin C, of which many semisynthetic derivatives were later produced, showing differences in antibacterial spectra and in susceptibilities to different beta-lactamases. In analogy with penicillin G, the side chain of cephalosporin C can be removed enzymatically to give 7-amino cephalosporinic acid, which can be acylated chemically to give new derivatives. The first successful semisynthetic cephalosporin was cephalosporin. Many others have followed. Most are effective only when given parenterally, but cephalexin and cefixime can be taken orally. Cefuroxime is not degraded by many common beta-lactamases and can then be used against pathogens that are resistant to many other beta-lactams. Like all beta-lactams, cephalosporins work by interfering with the cell wall synthesis of growing bacteria. As indicated, there have been both medical and commercial incentives for medicinal chemists to synthesize cephalosporin modifications. One has been to obtain new beta-lactams with wide antibacterial spectra embracing different pathogens. Another reason has been to find beta-lactams with an increased ability to resist beta-lactamases, which have been observed to increase in frequency among pathogenic bacteria. The latter goal has turned out to be very difficult to achieve, at least for more than a short time, since beta-lactamase evolution seems to have been able to keep pace with the ability of medicinal chemists to produce new derivatives. Now, sulfonamide. Sulfonamide, also called sulfa drugs, is the basis of several groups of drugs. The original antibacterial sulfonamides are synthetic antimicrobial agents that contain the sulfonamide group. Some sulfonamides are also devoid of antibacterial activity, for example, the anticonvulsant Sultham. The sulfonyl urase and thiazide diuretics are newer drug groups based on the antibacterial sulfonamides. Allergies to sulfonamide are common. According to data, the overall incidence of adverse drug reactions to sulfa antibiotics is approximately 3%, close to penicillin. Hence, medications containing sulfonamides are prescribed carefully. It's important to make a distinction between sulfa drugs and other sulfur-containing drugs and additives such as sulfates and sulfides, which are chemically unrelated to the sulfonamide group and do not cause the same hypersensitivity reactions seen in the sulfonamides. Because sulfonamides displace bilirubin from albumin, current citrus brain damage that is due to excessive bilirubin is an important potential side effect of sulfonamide use. Now, the functions. First, antimicrobial. In bacteria, antibacterial sulfonamides act as competitive inhibitors of the enzyme dihydropteroate synthase, an enzyme involved in foliate synthesis. Sulfonamides are therefore bacteriostatic and inhibit growth and multiplication of bacteria, but do not kill them. Humans, in contrast to bacteria, acquire folate through the diet. Sulfonamides are used to treat allergies and cough, as well as fungal and malarial infections. Now, quinolones. Nalidixic acid is the original substance of the group of synthetic antibacterial agents called quinolones. It was discovered in a synthesis program originally based on observation of the antibacterial properties of the oxoquinoline found as a byproduct of the industrial synthesis of the malaria drug chloroquine. This observation and a follow-up screening program occurred at the beginning of the 1960s and has produced a number of synthetic derivatives, which have become one of the most important groups of remedies in antibacterial therapy. They are distributed as inexpensive and efficient antibacterial agents, particularly against urinary tract infections caused by gram-negative enterobacteria. Examples include ciprofloxacin, inoxin, lomofloxacin, norfloxacin, and ofloxacin. Now, quinolones and their mechanism of action. First, they are bactericidal. They are effective against gram-negative organisms and some gram-positive organisms, alter DNA of bacteria causing death, and do not affect human DNA. Quinolones and their therapeutic uses. First, lower respiratory tract infections. Second, bone and joint infections. Third, infectious diarrhea. Fourth, urinary tract infections. Fifth, skin infections and sexually transmitted diseases. Now, aminoglycosides. Aminoglycosides are derived from various species of Streptomyces. In 1943, Salman Weizmann, together with his co-workers, discovered that a fungus, Streptomyces griseus, produced an antibiotic substance which they named streptomycin. Salman Weizmann was awarded the Nobel Prize in Physiology or Medicine in 1952 for his discovery of streptomycin. The aminoglycosides are bactericidal and work by stopping bacteria from making proteins. Aminoglycoside antibiotics are used to treat infections caused by gram-negative bacteria. Aminoglycosides may be used along with penicillins or cephalosporins to give a two-pronged attack on the bacteria. Aminoglycosides work quite well, but bacteria can become resistant to them. Since aminoglycosides are broken down easily in the stomach, they can't be given by mouth and must be injected. Generally, aminoglycosides are given for short time periods. Aminoglycoside group includes amikacin, gentamicin, kanamycin, neomycin, streptomycin and tobramycin. Aminoglycosides and their side effects. The major irreversible toxicity of aminoglycosides is ototoxicity, that is, damage to the ear and hearing. Among them, streptomycin and gentamicin are primarily vestibulotoxic, whereas amikacin, neomycin, dihydrostreptomycin and kanamycin are primarily cochleotoxic. Another significant concern with aminoglycoside antibiotics is nephrotoxicity, that's kidney damage. Renal damage is related to the accumulation of high concentrations of aminoglycoside antibiotic in the renal cortex. With this, we conclude our lecture. Thanks for watching.